Today we will cover complete SARS-CoV-2 spike protein, or in other words, complete glycosylation of SARS-CoV-2 spike protein. In our previous videos, or we can say in our last 2 to 3 previous videos, we have discussed only a part of spike protein of SARS-CoV-2, that is S1 part. However, this paper describes the glycosylation profile of whole spike protein of SARS-CoV-2. So this is the general figure that they have shown, and each site represent, and wherever they have found the glycosylation, they have represented them from top to down, and top one represents the N terminal and bottom one represent the C terminal, and we can easily see what are the kind of carbohydrate structures or glycans they are able to find at a particular site. In this figure, it is not clear the relative abundance of each glycans. However, they have considered that relative abundance in other figures. So this is just an overview. What are the types of glycans they are able to find in whole spike protein. And similar figure is shown in the panel A of the right side figure. In this, they have not only shown the glycans, but they have also represented them, that what are the function of these glycans. For example, this this gray color at 17, for this the function is not yet reported, whereas for the glycans at this N61 position, its function is to help the SARS-CoV-2 virus in the penetration to the host cell. And this diamond shaped yellow color represents the there is more this N149 helps in the mutated SARS-CoV-2 virus towards penetration, or in other form of infect, or in other way to infect within the host cells. And similar in this way, there are other symbols are also represented here. For example, this, you can say, white circle color represents the binding. So this type of glycans, such as in N165, 234 or 282, and this type of glycans helps in the binding of the SARS-CoV-2 virus to the host cells, host cell surface membrane. And this kind of glycans that are represented by yellow color, such as this N74 and 165, these glycans helps in increasing their infectivity and affinity whenever they are within the host cells, so that SARS-CoV-2 virus will use the host cell machinery more efficiently to produce their more spike proteins, at the same time infect the cells in a way that it will disrupt its normal cell operations. And this diamond shape orange color, that is represented here for N122 and 343 and 331 and 717 and 801 and 1074. These are the glycans whose infectivity got affected whenever there is a mutation in the virus. And this diamond shape pink color, that is represented here as N616 and 1134, and this helps in increasing the stability of the mutated SARS-CoV-2 virus whenever there is some mutation observed, whenever there is a mutation occurred in the SARS-CoV-2 virus. So these are some of the examples that are shown here of what are the glycans that are found in the SARS-CoV-2 spike protein and what are their functions. And this is the, you can say, spike protein ribosome receptor binding domain. This is the top view and this is the side view. And in this panel also they have shown where are the glycans that are located, so that we can understand in 3D how these glycans are playing its role, how glycan play its role towards helping the SARS-CoV-2 virus in infecting the uh, how this glycans helps how this glycan plays a role towards helping this SARS-CoV-2 virus uh, in to get into the cells. So this is the top view, and here we can see some of the glycans such as N234 and 122, 344, whereas and in this also the gray color represents the S1 domain, and this blue color represents and this we can say green color represents the S2 domain, and this we can say brown color represent the receptor binding domain. And if we go to this side view, we can see the other rest of the glycans are shown here, and here also the green color represent the S2 domain, and brown color represent the receptor binding domain, and this gray color represent the S1 domain. Now some of the glycans are missing in this 3D structure, as uh, they are not they are not completely visible either in the top view or in the side view. They must be in the they must be visible from some other view. That's why they are not shown here. If you need to understand about this whole SARS-CoV-2 spike protein glycosylation profile and how it works, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.